Hey everyone, and welcome to Science in Real Time. This is the show where we sit down with the people pushing science forward and talk about what's really happening behind the breakthroughs. Before we dive in, make sure to subscribe so you never miss an episode. We've got some incredible conversations coming your way. All right, let's get into it. Your presentation really was showcasing projects ranging from everything to cell painting to even neuronal outgrowth assay, which was incredible. So I was wondering, how do these techniques expand what is already possible within biology?
These are kind of like different complexities of screens, right? Like we can do a more basic kind of target screen, which depending on the system can be like really interesting, right? Especially like coupled with different counter screening techniques and specificity. Sometimes you don't really get that in some of the more basic screens, but we can kind of expand on it that way. But when you get into like more complicated models like the cell painting and the neuronal outgrowth, you get a lot more data from those screens, and you can actually kind of answer questions more of like, what are more kind of complicated profiles that we're looking at. We're not just looking at like it is there or it isn't there, the cells are alive or the cells are dead. We're actually looking at kind of the effect that compounds may have in the cells. So you actually get a lot more information in that, and the goal is in that case to have something that will be more successful when it translates out of the lab into more of a translational area setting, such as like any kind of clinical trial or animal model or larger scale model that people start to take the leads that we identify and bring them into.
As I was hearing in your presentation, the idea of miniaturizing cell painting to a 1536 well format is incredibly fascinating. I was wondering, what were the biggest challenges and breakthroughs in making that scalable?
Typically everything that we do is 1536, which gives us a lot of breadth, and so it was important to try and attempt to do so with the cell painting as well. Victoria was the intern that came in and did the work this summer, and she did a really great job. We did kind of do some playing with like our liquid handling. When you work in 1536, you can't manually do any of that by hand, so you have to use liquid handling instruments in order to add liquid to plates or cells to plates or dye to plates, and as well as removal. So we do have a couple of techniques that we've kind of adapted for these kind of situations in order to work in 1536. So Victoria did a little bit of optimization with like dye concentrations as well as like pressure on the liquid handling system, so that as to wash off cells at the bottom of the plate, and some other kind of optimizations that she did for 1536, and she did a really, really great job. So.
And I've also seen that you worked very closely with Aracelis and Endeavor platform, naturally. I was wondering, what did that integration enable that wasn't possible before?
It's really like a modern instrument, which is really nice. And so you get one, like the software is really user-friendly. It's really easy to kind of understand what things that need to be modified in order to get really an excellent image. And that makes it much, much easier. The kind of speed at which things get done, you really benefit from that, because let's say you image a plate and you're like, actually I don't think I like that so much. It's really not a big deal to just go back and do it again. And you can have like multiple exposures. You're saying like, I'm really trying to find the best exposure time for like this set of dyes. So you can try like a couple different conditions, and you can even compare things like an analysis later on, which is really, really helpful. And that really gets you the kind of like best assay and like best data.
Absolutely, especially with how quickly things are moving, being able to generate that data, the quicker that you can, the better, I can imagine.
Yeah, no, for sure. I mean, we move really fast with stuff, and we're trying to, get things in and out of the door so that we can have the most capacity like that we can manage, right? And the speed of the imaging acquisition really helps with that, and it really helps us optimize much faster. So.
Absolutely. And now I want to switch gears a little bit from targets to phenotypes. I want to talk a little bit about the power of integrated screening. That was very much part of what you were talking about earlier today. And during your presentation, you were mentioning how HCS enables really information-rich assays. I was wondering, what kind of biological questions can we now answer that were previously out of reach without this technology?
It's really, like you said, it's information-rich, so you can ask, more complicated questions versus just kind of like there or not there, live or not alive. You can do localization, you can do kind of like intensity of spots, number of spots, you can count like spot detection, you can look at like morphological profiles, so the cell painting, are we like drastically changing the size of cells or the size of nuclei? These are all like more complicated questions that you can ask about your drugs. And ultimately what it does is it will lead to a better drug candidate at the end of your screen, right? One of the things that we kind of fight all the time is having like non-specific hits or like off-target effects that happen from compounds. And so when we design screens, we want to do the best we can to minimize any kind of like non-specific, off-target, whatever compound that we come in contact with, and really like focus on the ones that will be like truly efficacious when they come out of our lab. And so when you do high content, you're able to really get more information about the compounds that you're working with, and that really leads to a better lead ID or lead identification down the road.
For those building screening programs today, like you have been, what's the most important when integrating imaging, automation, and data analysis, in your opinion?
I think there's like several things that are pretty important. I think one, you really need to like take into context your like biological question when you look at your images, right? So it's really easy to just say like, oh, we see fluorescence, it's good enough, right? Which is not typically the case. You really need to think about like, what are the questions that we're asking? Does this make sense in our model? You know, is this what we're wanting to do? For automation, I would say like washing is really difficult when you get to automation. So there's things like a blue cat washer, people use that, which has, advantages and disadvantages. So if you're gonna look to move to automation, I usually try to focus on dyes that are friendly for automation, and that is like a really kind of like a game changer for transitioning from like a manual platform to like an automated platform. And then for the actual image analysis, I think people underestimate like just the capacity of like data that you can generate via images, right? Like you generate a lot of data from just like taking like really nice like high resolution images, but you have to figure out like, how am I gonna work with those? Like, how am I gonna manage those? Like, what's the pipeline in which I can do analysis? And do I have the computing power in which to do that analysis? Those are like questions that you kind of ask yourself beforehand, before you generate like the terabytes of data or whatever that you get from taking all the images that you wanna take. You have to ask yourself like, okay, do I have the computing capacity and like the storage capacity to handle this type of analysis? So I think those three things are like kind of key when you're like looking at a high content screening. So
absolutely. I mean, as somebody who deals with a lot of data, naturally this is a little different because it's a lot of videos and stuff, but certainly being sure and making sure that you have space in your computer is crucial.
That's really important.
Yes. And now I wanted to kind of like dig a little bit deeper into this AI automation and what, in your experience, do you see as the future of discovery? So the first thing that I was seeing during your presentation is the remarkable examples that you were giving of auto HSC, specifically identifying outgrown modulators using AI. And I was wondering, how does this approach change the pace or accuracy of discovery?
Yeah, I think it does a really good job of kind of like taking some of the conventional rules away from things that maybe like don't fall into conventional buckets, right? Like we do a lot of like cell segmentation, and that's kind of like the classic high content way of doing analysis, right? But neurons specifically are really hard to segment. Like you can ask anyone probably at this conference how difficult it is to segment a neuron, and they will, kind of tell you about it if they've tried, for sure, right? But with the work that we were doing in Vicky with their auto HCS toolkit, they actually don't need to like particularly segment. They just do kind of like bulk comparison of like that well with another well, and that removes that kind of hurdle from that analysis approach. And so I think, that's gonna like make a big difference going forward about how we think about these problems and how we like go about like trying to like quantify different cell types and things that are just like a little bit abnormal and that don't fall into this like traditional like globular cell, right? And we have more tools to do so.
When you think about integrating AI, specifically with imaging workflows, I'm wondering what excites you most and what do you think that still needs a little bit of refinement?
It's gonna be really exciting to just be able to do something really in real time where you like take an image and you have some kind of segmentation or whatever that you've maybe pre-planned beforehand, but that does like just a spectacular job and there's not much tweaking from you. I think a lot of people spend a lot of time like tweaking their segmentation algorithm or like trying to like edit stuff just to like get it right, and then they're like, oh, we didn't get that nuclei, or maybe is that one nuclei or two nuclei? You know, you have this like internal debate with yourself, and there's a lot of like user error in that actually too, right? Like you as a person, you know, I think earlier there was a talk, they talked about like comparing the AI function to like undergrads doing their own kind of drawing of stuff, right? So taking that out of the question and having something as an AI that like comes into the space for that, I think that's gonna be really exciting, and it's gonna be a nice automated way to go about image analysis. And I don't know, I'm not like the leading AI person, but I would say that like, yeah, it would be nice to just be able to more ubiquitously apply these things. A lot of times you kind of have to be an expert in AI or like understanding like machine learning to be able to apply these things. And there are tools like coming out, like companies have tools that have like the AI integration. The Clairvoyance has like an AI, like a deep neural network, right? So there are tools in which it makes it a little bit more user-friendly for your kind of average lab user to do AI. And I think like more of that and more like complicated versions of that will probably be like really impactful for, you know, scientists today.
Absolutely, I certainly agree with that because it doesn't necessarily is the most intuitive.
Yeah, I know it's not always. Yeah, sometimes there's like a large learning hurdle between like you know, like average scientist who does really great cell biology to do really complicated image analysis.
Companies like Araceli are really enabling scientists to visualize biology at scale. And I'm wondering, how do you see technology partnerships like the ones that you have had with Vicky and Araceli as well, shaping the next generation of discovery?
From the work that we've done with Araceli, like y'all have been really good about kind of like taking our feedback and then like really improving like and doing more, you know, from that, right? And I think that is like really exciting. I think that companies that are enabling that sort of stuff, when you have conversations with like real users, you can really see like what they need and like what is like practical in their hands, right? And so like, yeah, like there's several software things that we've like discussed with some of the team, like we really want this button, you know, and that it really makes a difference in the end, right? And then it enables like a functionality you probably didn't even really think of, and that is really impactful for, you know, your everyday kind of user situation. And so, yeah, I think that like that relationship is really important, and like having like that communication with like the people who are actually doing the science and the people who are creating products and like instruments for people to use. I think it's a like really crucial relationship to build and grow upon. So
I agree, and ultimately you folks are the ones using the machines, so we want you folks to be able to use them to the most of their capabilities. So that makes a lot of sense. And you know, looking forward, do you think data, model systems, or even analytical algorithms will be the biggest drive of innovation in the next few years?
I do think technology, like having like physical technology, like physical, you know, microscopic technology, like how we design our microscopes, how we're using different lasers, how we're like doing different kind of cameras. I think that all has its own place. Like I think that is also going to be important. I know now is kind of the age of like the AI boom, right? And so I can't disagree that it's probably going to be a big driver of innovation in the future. But I don't like think we should discredit like having a physical good instrument like that takes nice pictures. Like that is really important, and that is probably going to always be important. And there's always like engineers and stuff. They're always coming up with really great kind of like improvements upon those. And so yeah, like having like a really good camera, that really kind of makes or breaks your high content imaging.
The point of just because we have AI, we should not forget about the things that we have been using already, and not putting, you know, some TLC into them as well, making sure that they're also in a good condition. And now I want to switch gears a little bit about yourself, behind the science, the motivation and the mentorship. I know that you have wore a lot of different hats in all the places that you've been, and you have been able to really have a very successful career. And I was wondering what originally sparked that passion for discovery science?
Yeah, okay, so my first kind of science experience was in undergrad. I joined, HHMI has like a C-phages initiative, and I was part of that. And honestly, that was like one of my first science experiences, and it was like really positive, right? Like I wasn't planning on doing research as, you know, as an undergrad, freshman undergrad, I wasn't really planning on doing research as my career, but like that experience was really great. I really enjoyed it. I liked this idea that like I'm gonna do something that no one else has done before, that I get to like solve problems and think about things that no one else has done before. And so yeah, like I think that like these educational programs that they offer for undergrads, like with integration and research, they're really important because I think that opened my like world to a bunch of different possibilities that I really, at that point, I had no idea. So that was one of my good, like kind of like, ooh, this is really fun. Like I get to go out in the world and like find something new. That's so crazy, you know? It's such a good experience to have, so.
Out of all the projects and experiments that you have led, is there one in particular that taught you most about perseverance or even creativity in research?
If I kind of go back to like my PhD work, that was kind of really kind of a game changer. I mean, most people will say like their PhD kind of changed their like perspective, or their experience in grad school changed their perspective on a lot of things. I think, yeah, like the work that I did in my PhD was just like basically the biggest kind of like logic puzzle that you've ever seen, right? So biochemistry innately is very, not visual, very different from high content imaging. There's like, it's not, you can't see anything actually. You just kind of like throw stuff at water in a tube, and then you kind of put it in a reader and kind of see what happens on the other side. And then from that, you have to make inferences about what's happening that you physically cannot see. Yeah, I think that required a lot of me to be able to kind of imagine, okay, if I do this, this is the result, what does that mean, you know, and how does that change like my perception of what may be happening in the water in this tube, right? And there's a lot of instances where I had to come up with like really creative like combinations of substrates and everything in order to like get a better understanding of you know what was going on in the system that I was looking at. So I really liked it. It was very creative. It was very kind of like, okay, what am I gonna do? Like, what am I gonna try next? You know, type thing. And it was really rewarding in the end. Honestly, it was really rewarding. So I'm glad that I did it.
I am very glad that you did it as well, because you're very good at it. So I'm very, very glad. And how do you approach mentorship or inspiring young scientists to be able to enter this rapidly evolving field? You have had plenty of experience since you were in undergrad. So I'm wondering, what are those things that you do to make sure that you continue helping propulse the next generation of scientists?
Yeah, I think it's really important for kind of young scientists to have some excitement about science, right? Like that kind of like curiosity and excitement for discovery really like needs to be kind of ingrained in them, right? Because if they have that little seed of like excitement about like learning something and, you know, being able to discover new things, it really motivates them to do a really great job. And I think that's really pivotal in like in a trainee or like an undergrad or an intern. If they have those kind of internal motivations, and they're going to do excellent in the lab, and you can kind of like pose the right questions for them to really grow in the environment that you're like providing them. So yeah, I think that is like a really big thing. I do really like the creative aspect of science. I tell people that I feel like science is structured creativity. Maybe in another life I would have been like an artist or something, but in this life I really do like a good structure, and I like that science like affords me the ability to be creative, but like in a, you know, the laws of physics, right? So I think that it's, you know, it's just pivotal like for anyone who's kind of like new to science, like they find a little bit of like that joy and like that excitement as well, and I think that pushes them to you know, be motivated and to like really improve themselves and improve their science.
To wrap up, I was wondering, for listeners that are very interested in learning more about your work, yourself and even UF Scripps as a whole, or even high content discovery, everything that you folks are doing basically. I'm wondering, where can they find your work? Where can they go?
Yeah, absolutely. So I have my LinkedIn profile. It's just my first last name, Claudia McCown. So I have a LinkedIn profile. It has publications for me, and then I have connections with the people at Scripps. Scripps also has a website, and the High Throughput Screening Center has a web page as well. It lists all of our publications that we have done recently, and can see the different assays that we've been able to do. So yeah, I think either of those resources you can probably get some good background. So
that pretty much concludes our interview. I want to thank you so much for joining us. Your background and all the work that you folks have been doing, it's incredibly amazing, and you folks are really pushing the needle of discovery, and it's been an honor to be not only able to have you here, but also be able to see you in action with your presentation, talking science. You did wonderfully, and I feel very lucky to have been conducting this interview.
Thank you. Yeah, thank you for having me. It has been great.
Thank you so much for tuning into Science in Real Time. We're really glad that you were able to spend time with us. If today's conversation sparked questions or ideas, we'd love for you to keep it going. You can connect with our guest and the Science in Real Time team using the links in the description box below. And if you enjoyed this episode, don't forget to like, subscribe, and share with someone who loves science as much as you do. Until next time, thanks for watching.